Welcome to today's session. Today I will discuss about different staining methods. Common staining methods are gram staining, gel Nelson staining, Albert staining, Giemsa staining, Leishman staining, negative staining. Another name is India ink staining. From here, I will describe gram staining, gel Nelson staining, and Albert staining in details. First, come to gram staining. First step is preparation of smear. At first, take a clean glass slide and put a drop of normal saline at the center. Mix a small amount of supplied specimen with normal saline and make a homogeneous mixture, and then spread over the glass slide. Then let it air dry. Then fixation of smear. Usually we do heat fixation. This prepared smear is passed through a flame 3 to 4 times for heat fixation. Fixation is important because during a staining procedure we will wash this smear different times, and during this washing the organism may be washed out. That's why fixation of smear is important. Then primary staining. Primary staining in gram staining is done with crystal violet. The smear is covered with crystal violet for 1 minute and then wash with tap water. Next step is mordanting. Mordanting is done by Lugol's iodine or Gram's iodine. The smear is covered with Lugol's iodine for 1 minute and then again wash with tap water. The next step is decolorization. Decolorization is done by acetone. The smear is washed alternatively with water and acetone until it becomes colorless. Then counter staining. Counter staining is done with diluted carbolfuchsin. Color of this diluted carbolfuchsin is red. This smear is covered with diluted carbolfuchsin for 30 seconds and then wash with tap water. After washing, the smear is air dried and then focused and observed under microscope. In case of gram-positive bacteria, peptidoglycan layer is thick. So this peptidoglycan layer is not completely decolorized by acetone, and they retain a stain of primary stain. Primary stain, that is crystal violet. That's why gram-positive bacteria takes violet color on gram staining. In case of Staphylococcus, they are arranged in cluster, and in case of Streptococcus, they are arranged in chain or pair. On the other hand, in case of gram-negative bacteria, peptidoglycan layer is thin. So, this peptidoglycan layer is completely decolorized by acetone, and when counter-stained by carbolfuchsin, gram-negative bacteria take red color. That's why gram-negative bacteria are red in gram staining. And in gram staining, what will be the color of background? When the smear is decolorized, the background also become decolorized like this. But when counter staining done with red color carbolfuchsin, the background will take red color. That's why background of gram stain will be red. Sorry. And this is Staphylococci. These are Streptococci. Now come to gel Nelson staining. First step is preparation and fixation of smear. This smear preparation and fixation are similar to gram staining. Then come to primary staining. Here primary staining is done with heated, fuming, concentrated carbolfuchsin. Every point is important: heated, fuming, and concentrated carbolfuchsin. Here heated carbolfuchsin is used because mycobacteria has thick mycolic acid layer in their cell wall. This mycolic acid is a lipid substance, and for better penetration of this layer, we use heated carbolfuchsin. We will cover the smear with heated concentrated carbolfuchsin for 5 to 10 minutes and then wash with tap water. Then decolorization. In case of Mycobacterium tuberculosis, we use 20% sulfuric acid for decolorization, and we cover the smear with 20% sulfuric acid for 1 minute and then wash with tap water. But in case of Mycobacterium leprae, we use 5% sulfuric acid, then it is called modified Zell-Nelson staining. And in case of Nocardia, we use 1% sulfuric acid. In Zell-Nelson staining, there is no step called mordanting, because here we use heated carbolfuchsin. This heat serves the purpose of mordanting. After decolorization, the next step is counter staining. Here we use Loeffler's methylene blue for counter staining. The smear is covered with methylene blue for 30 seconds to 1 minute and then wash with tap water. After washing, the smear is air dried and then focused and observed under microscope. In case of Mycobacterium tuberculosis, they have thick mycolic acid layer in their cell wall. That's why they resist decolorization by sulfuric acid and retain the color of primary stain. That means retain color of carbol fuchsin, which is red color. That's why Mycobacterium tuberculosis are red on gel Nelson staining. And there will be some granular appearance called beaded appearance, because due to disruption of mycobacterial cell, there will be uneven distribution of this mycolic acid. That's why they produce this granular appearance, that is called beaded appearance. And after decolorization, when we use this methylene blue for counter staining, the background will be blue. Background will be blue. But in case of Mycobacterium leprae, we will decolorize by 5% sulfuric acid. And this Mycobacterium leprae will be short and stout and arranged in bundles, and they will be uniformly stained and there will be no beaded appearance. Now come to Albert staining. Preparation and fixation are same as before. Then we will cover the smear with Albert stain 1. Albert stain 1 reagent, which is a mixture of toluidine blue and malachite green. The smear is covered with Albert stain on reagent for 2 to 3 minutes and then wash with tap water. After that, the smear is covered with Albert stain 2, which is also called Albert iodine. The smear is covered with Albert stain 2 reagent for another 2 minutes and then wash with tap water. After washing, the smear is air dried and then focused and observed under microscope. Albert stain is used to see the bacteria named Corynebacterium diphtheria. On Albert stain, these Corynebacterium diphtheria are V and L shaped, deep green bacilli. V and L shaped deep green bacilli with bluish black metachromatic granules. These are bluish black metachromatic granules. And the background will be light green. This background will be light green. And while some bacteria are arranged closely and they produce Chinese alphabet pattern. Chinese alphabet pattern. These are three staining methods: Gram staining, Gell-Nielsen staining, and Albert staining. These three staining methods are very important, and Gram staining, Gell-Nielsen staining methods are important for both OSCE and practical examination. And hopefully this will be benefited for the students. Thank you. Thank you for hearing. And next day I will come with new topic.